Bismillah Rahman Rahim 5090 June 25 question paper 4-1. I hope this is going to be an interesting video before your exams takes place, the November 25 exam. As you all must know that this paper is for 1 hour and you must be able to complete it in about 45 minutes so that you have 15 minutes for revising it and then rechecking it. Because the present generation Gen Z makes about 5 to 10 marks of careless mistakes. So please, I would request you to finish this paper in 45 minutes and the last 15 minutes look for your careless mistakes and rectify them. When attempting paper 4, which is an alternative to practical, you must read the whole question first and then start attempting it because then you could be making some mistakes in just doing parts of it. Now, reading the question 1 first, I'm not going to read the whole question, but I'm going to do parts of it. But when you're taking the exam, you must read the whole question. A student investigated the effect of gravity on germinating seeds. 8 pea seeds were used. As they germinated, the pea seeds first produced radicals, roots. The length of the radical can be measured between the dotted line as shown in figure 1.1. So you have to measure this between the dotted line, this area, with a ruler. The student attached 4 of the seeds with germinating radicals to the cork base on each of the 2 electric motors as shown in figure 1.2. The cork was kept wet during the investigation and the apparatus was kept in the dark. So direction of movement and then electric motor to turn the cork base. So this is A and this B. So the student then switched on motor A so that the cork base rotated very slowly. So A was rotated very slowly for 2 days, turning in the direction indicated by the arrow, this arrow. Motor B was not switched on, so the cork base did not rotate. So this was not switched on and this was kept straight and so it was not rotated. Then we say figure 1.3 shows the p radicals after 2 days. So direction of the movement was like this and these are like this and these have grown like the seeds, you can see they have grown down while these have grown a little longer but they are in the same direction, they are horizontal because you see gravity was pulling on all sides, so that's why they are growing horizontally because it was rotated very slowly for 2 days, so each side got the effect of gravity. This was only getting gravity on the lower side, this side was only getting the effect of gravity. So then of course that is why this one grew, it was going to curve downward, so this is the way it was going to curve because the effect of gravity is on the lower part of the radical. Then part A says figure 1.4 shows the actual size of the 4 germinating peas from A after 2 days. So you've got them. Then it says figure 1.5 shows the length recorded in millimeters in the student's notebook for the 4 radicals from B after 2 days. So one is 35, 40, 33 and 36. You are going to measure and record the length of each radical from A, the distance between the dotted lines in figure 1.4. In the space below, construct a table in which to record your measurements from A and the student's measurements from B. Then it says measure and record in your table the length of the radical shown from A in figure 1.4. Then record the student's measurements in your table for the radicals from B in figure 1.5. That's another 1 mark. Then calculate the mean lengths of the radicals from A and B and record them to 1 decimal place. So mean length of A and mean length of B. Now let's complete the tables. Now this is the initial table which you are supposed to make. So single table with columns and rows drawn with ruled lines. You got a mark for that. Header for seed in the column. So you written seed 1, 2, 3, 4. And header for the height in millimeter in the second column. So you can say the length or the height. But this millimeter should only be written in the heading. You should not write millimeter in everything here. If you write it like this is a -1. Remember, you can't do this. You can't write this in every line and say millimeter. You have to just write millimeter in the headings. Always. This is the rule, I think, in bio and physics and chemistry everywhere. And then the header for A and B. So you're going to put this here and you got a header here for A and a header here for B. So this is how you are supposed to make the columns, and this would of course now get you your 4 marks, because you're going to measure two different ones, A and B. So you had to give the length in millimeter, and then you had to give me A and B. Then it says measure and record in your table the lengths of the radicals from A in figure 1.4, and this is for 2 marks. This is figure 1.4. So seed 1, and you measured it, it was 33, and this was 36, but it's anything between 35 and 37 was correct. This is 35. What is 35? Millimeter. And this was, say, 38 or 39 or 40 was even correct. So you measured them here, you wrote them here, but then you've got to put them back in the table. So you wrote these here and you get the marks, the 2 marks, because you measured them and then you wrote them here. So you get 2 marks for that. Then it says record the student's measurements in your table for the radicals from B in figure 1.5. This is figure 1.5. In this the readings were 35, 40, 33 and 36. So just writing these, because those you just had to copy them from figure 1.5. So writing that is only for 1 mark. So you got a mark for that. Now it says calculate the mean length. So you calculated the mean length. It was roughly 35.8, because it depended what you wrote for that. And this was 36.0. 0. 0.0 is important because it said 1 decimal place. There's a difference between significant figures and the difference of decimal places. There's two different things. Please remember to be very clear on that. Now part 4 says use figure 1.3 and the mean lengths in 4 to suggest what the student should conclude about the response of the radicals to gravity. Use figure 1.3. This is figure 1.3. So you use this figure 1.3 to explain all this. Now please remember, when it says suggest what the student should conclude, this is part 5 of the question. Radicals in B curve down, and in A they do not. Radicals in B curve down, in A they do not. Gravity acts on B, or you can say B shows positive gravitropism. What does the rotating drum do? It cancels the effect of gravity. But it keeps on rotating it, so every side gets the effect of gravity. And there's a very little difference in the overall lengths. It's nearly the same. So it was for 3 marks. It says to suggest what the student should conclude about the responses of the radicals to gravity. So this is how we would word it. Radicals in B curve down, in A they do not. Gravity acts on B, or you can say shows positive gravitropism. And rotating drum cancels the effect of gravity, and little difference in overall lengths. Then we come on to the B part of the question. Suggest why the student should repeat the investigation several times to obtain results for more pea seeds. You see, we can then identify the anomalies. You know, this word anomalies means if there are any sort of, there's some seeds may not germinating properly, maybe they are damaged, maybe they have some sort of a disease on them. So anomalies, or the other word that we use is outliers, outliers. And 4p is a very small sample. Or you can say to make the results more reliable. When you repeat an experiment, R repeat, R reliable. Reliable means that now we have 5 or 6 readings and we can say, okay, this is what is actually the case, what happens. So this is what we say is, when we repeat the experiment, is the results more reliable, or you say identify the anomalies. I've talked about this in previous papers as well. Now coming to the C part, suggest why the apparatus was kept in the dark during the investigation. Why? Because we didn't want another factor affecting the growth, because light does also can cause it to grow in a certain direction or away from light. So light does not affect the results. Gravity is the only factor that is being investigated. And the reason is their roots are usually grow in the dark and they're underground, so they're not exposed to light. So these were the reasons why we kept it in the dark. So this is how we would word it, that light does not affect the results and the gravity is the only factor being investigated, and the fact that roots normally grow underground and they have no effect of light on them. Now coming to the D part of the question, another student investigated the growth of the shoots, plumules of germinating pea seeds for 8 days. The student's results are shown. So time and days and the mean height, and you can say the unit is written always in the heading. So mean height of the shoot. Now this first column is always the independent variable, and this is always plotted on the x-axis. This is the rule that we follow. That's why the mnemonic is FIX. First column, independent variable, x-axis. And then mean height of shoot in millimeter will go on the y-axis. This will go on the y-axis. This whole thing will go on the y-axis. So if you just label the axis, you actually get 1 mark. And you haven't drawn anything, you still get 1 mark. And then I get very angry when I check papers and I see that some of you have not even bothered to label the axis. And that's something that's totally a careless mistake. I do not respect you for it. I do not even uh, appreciate, I feel all the money that your parents have spent and you wasted it by just doing a careless mistake of not labeling the axis. It's very sad for me. It's very sad if you do such a thing. And some of you do it. I'm sad, but you do it. So mean height of the shoot in millimeter goes on the y-axis and time in days goes on the x-axis. You can see the time in days is 02468. So please spread it on the grid. So this is how I've labeled it. You label it every 2 centimeter, 0, 2, 4, 6 and 8. Now you see what are the scale that you're going to take on the y-axis. You decide that after looking at the figures on it. Now you can see that the y-axis has got to be spread up to 16. So you have 3, 6, 10, 13 and 16. So I've decided to take it because I like to keep the scale a bit easy. So 10 small squares is equal to 5. Yeah, that means 2 centimeter is equal to 5. You see, this is 2 centimeter. Remember, this is 2 centimeter. So this is equal to 5. So I've decided to take 0, 5, 10, 15 and 20. Now I have to plot it. For 0, I'm going to plot here. 0 time was 3 millimeter. So I'm going to plot it and show you how I plot the different points. Now you see labeling the axis is 1 mark, then choosing the scale to use the whole grid is another mark, and then it says time on the axis and both axes fully labeled with the units, you got a mark. Then linear scale and values at origin and good use of the grid. Then 5 plots correct, you got another mark, and then the points joined with ruled lines and no extrapolation. No extrapolation means no extending it. Just join the points, the first, the second, third, and the fourth, and the fifth point, and that's it. Don't extend it either this way or either one, this side on the left side or on the right side. Now, very important that you see the dot must be a dot, not a blob. This is a blob, a dot, and then the next dot is another dot here. So you must connect it very carefully. It should not go. The line should not go below the dot. Either you use a dot and you circle it, or you use a cross. So this is up to you, but please do not circle the cross. Either you use a dot. Please remember a dot, I said, not a blob. This is a blob. So your pencil should be very well sharpened. Then the last question says use your graph to estimate the mean height of the pea shoot on day 5. So day 5 would be somewhere here. So you got to figure that out. So you draw a line on it. It says show your working on the graph. So you draw a line here and you extend it here, and then you take it to the side. I might not have done it very correctly. So now, I mean, wondering it, I'm saying, how would you get your 3 marks? It's a very easy 3 marks. Number 1, correct values from the graph. Number 2, graph working shown. You've shown this line. You've shown this line, which I'm now coloring in black. So you've shown this line. Some of you will just not bother to draw this line. And then, of course, the right units. 11.5 you'll write, but you won't write millimeters. Careless mistake. Pathetically chungar kids. Yeah, that's what you do if you do careless mistakes. I have no respect for you. No respect for you. Question number two, figure 2.1 shows part of the shoot of a pea seedling. Make a large drawing in the space below of the pea seedling as it appears in figure 2.1. So just do a drawing and please do not worry. My drawing is not very good and all that. Just don't give me all that crap. Just it has to be clear and clean lines drawn with a sharp pencil, no ruled lines, no shading. Minimum size should be at least 90 millimeter, and that I tell you a very easy way to do it is you cover this whole space till the where the marks are written. So you cover this whole area, nearly the whole area, near 3/4 of the whole area. So it should have been 90 millimeter. This is the minimum size. It can be more than that. And then detail of stems, double line, one main stem, three branches, number of leaves, minimum 7, bottom left lower branch, two leaves delimited, and correct position of the tendrils, which are 3. Now, if you look at my drawing, I know my drawing is not very good, but the main thing is that you have to draw what you're actually seeing. So clear and clean lines drawn with a sharp pencil, no ruled lines, no shading. Minimum size 90 millimeter. I haven't measured mine, how much it is. Detail of stem, double line, one main stem. So you have to draw a double line here. I would lose a mark for that. So double line, one main stem and 3 branches, right? So double line, one main stem, 3 branches, number of leaves minimum 7. So I have 1, 2, let me just put dots on it. 1, 2, 3, 4, 5, 6, 6 and 1, 7 here. So I've counted 7 at least. So 1, 2, 3, 4, 5, 6, 7. 7 leaves minimum. Bottom left lower branch 2 and bottom left 2. This is the one. This is the one. The bottom left one. And they are these 2 leaves. And then the correct position of the tendrils. These are the tendrils. These are the tendrils. So this was 5 marks. This was for 5 marks. A lot of marks for a drawing. So drawing should be as exact as you can make it. Straight, simple lines, sharp pencil, no ruled lines, no shading. Then part 2 say the leaves of the pea shoot are green. Describe how you to prepare a leaf from the shoot and carry out a test on it to show that it contains starch. And this is 3 marks. So you dip it in boiling water. Then you boil it in ethanol. And then you just dip it in water to soften it. And then you add iodine solution, and if starch is present it will turn blue black. So basically you got to give me 4 sentences which are very simple sentences. Dip in boiling water, boil in ethanol to remove the alcohol, to remove the chlorophyll, I'm sorry, to remove the chlorophyll. I'm also becoming changad, I think, like the Gen Z. Use of water, so you dip in water to soften it and then you add the iodine solution and it turns from brown to blue black, and that shows starch is present. Question number 3. Urine tests can be used to diagnose diseases. Indicator paper strips are dipped into a sample of urine. They detect substances excreted in the urine sample that are not normally present in the urine of a healthy person. 3 examples of indicator paper strips are diastics to detect glucose, albustix to detect protein, and ketostics to detect ketones. Patients with diabetes excrete glucose and ketones. Patients with nephritis excrete proteins, and patients with malnutrition excrete ketones. Table 3.1 shows the results of tests on the urine sample of 4 individuals, D, E, F, and G. A tick indicates a positive test and a cross indicates a negative test. State and explain which individual is healthy. Which one is healthy? F is healthy. Why is he healthy? Why? Because all is negative. He should not be having all this. The diastics is also negative. The albustix, the ketostics of F, all tests are negative. Positive means he has a disease. There's only 1 mark for that. And then it says state and explain which individual has diabetes. E. Why E has diabetes? Because positive for glucose and ketones. E has diabetes. Why E has diabetes? There are 2 marks for this. So E got you 1 mark and positive test for glucose and ketones. Positive test for glucose and ketones. You can see what was E for. Albustics is for, that is negative. So there's no proteins in the sample, but the person is positive for glucose and ketones. So glucose and ketones are positive. So that is why this person would have diabetes. Now coming to the last part of the question, the pH of urine in healthy humans is normally 6.5. The pH of urine can be measured using universal indicator paper strips. A scientist wants to determine whether the pH of human urine is affected by the volume of water drunk. For her investigation, she selects 20 healthy female volunteers of similar age and weight. Plan a method the scientist could use for the investigation. So it says, urine is affected by the volume of water drunk. So first of all, you'd have to have a baseline. Like for instance, I say, well, okay, I'll do a test when you come into A1. I will do a basic test of O-level biology, cell structure, biological molecules, and enzymes. So I then know how much you know of the O-level. Though you have your CIE grades, but I want to get a baseline. So I do a very simple test and I say, okay, everybody does this test for 20 marks and you do an MCQ. So first of all, check the pH at the start of the investigation for the 20 people. Then you give them different volumes of water to drink. 10 ml, 20 ml, 30 ml, 40 ml. Now same time interval between drinking and measuring the pH. So okay, you had the water at 8 a.m. in the morning. So now at 4 p.m. we are going to check your urine. Then same diet. They should have the same diet, the same exercise. They must be in the same temperature because if they're in a hot room, then they're sweating a lot. And then you measure the pH values of the urine samples. You look at the color change on the universal indicator paper compared to the standard chart or use the pH meter. And then you calculate the mean pH for different volumes of water and then you look for the relationship. We don't ask you what the relationship is, we just tell you, okay, how will you do this experiment? So don't get confused and start writing, okay, this will happen, that will happen, that. We don't expect you to come up with that. We just expect you to tell us how will you do this practical. Now, whenever there's a question on this plan, a method the scientist could use for investigation or something, you always think of it actually, what would you do if you were doing this in the lab? So first you need to check the pH at the start of the investigation for the 20 people because what are you going to compare it with? Because you're going to give them drinks of water, different volumes of water to drink, and then you're going to say, okay, that changes the pH. So let's do some investigation before that. Before we start it, we should have some baseline on which we started. So check pH at the start of the investigation for the 20 people, then 5 people get 50 ml, 5 get 75, 5 get 100 ml, and 5 get 125 ml. The 20 people, I've divided them into 4 groups. So one will be group A, this will be group A, this will be group B, this will be group C, and this will be group D. And each 5 of them are giving different volumes of water because you said range of volumes of water to be drunk. And then you're going to say, okay, you're going to have it at 10 a.m. in the morning. At 3 p.m., you're going to collect the urine samples and then check the pH because it must be for a fixed time. You can't just tell them to come the next day or the day after or 3 days later. Then you keep them on the same diet and or the same sort of exercise or the same environmental temperature because you see, if they're sitting in a very hot room and some of them are in a cold room, so the person in the hot room is sweating, that is going to change the volume of the urine. And then you, after this time, specified time, could be 5 hours or 6 hours, you've got to give me some time frame, can't be after 5 days. So measure the pH value of the urine samples. How do you going to use it? Either you use a universal indicator paper or you use a pH meter. And then you calculate the mean pH values for different volumes of water and you see in group A, group B, group C, was there some relationship? So by changing the volume, was the pH affected or not? Maybe it's affected, maybe it's not affected. You're not supposed to know that. You're not supposed to come up with some sort of a conjecturing and some sort of analysis. We don't expect that from you at O levels. So that completes this paper 4-1, and I hope this is helpful for you because very soon some of you taking the November exam. So best of luck and best wishes.